Human cells can break down glucose without a single drop of oxygen, but this process, glycolysis, only yields a net of 2 ATP molecules per glucose. For an organism with our baseline energy demands, that thermodynamic math is a death sentence. To extract the volume of energy actually required to keep human tissue alive, the cell must shift away from anaerobic processes and initiate aerobic respiration. We can see exactly why this shift is vital by looking at what happens when it is blocked by cyanide. Cyanide doesn't interfere with glycolysis. Instead, it binds irreversibly to a protein complex in the electron transport chain, specifically preventing oxygen from binding. Without oxygen acting as the final receptor, aerobic ATP production drops to zero within minutes. Aerobic respiration operates as a highly interdependent engine. If you jam the final sequence, the entire energy-producing system backs up and starves the cell. The physical setting for this massive energy extraction is the mitochondrion. The interior is divided into specific spatial zones, the dense inner fluid known as the mitochondrial matrix, surrounded by a highly folded inner membrane called the cristae. This strict physical compartmentalization dictates exactly how chemical energy is extracted, transported, and ultimately converted into a usable form. The process begins when pyruvate, the end product of glycolysis, crosses into the mitochondrial matrix. Here it encounters the pyruvate dehydrogenase, or PDH, complex. This multi-enzyme structure serves as the one-way gateway into aerobic respiration. Within this complex, the 3-carbon pyruvate is irreversibly cleaved. 1 carbon is released as carbon dioxide waste, and the remaining 2 carbons attach to coenzyme A, forming acetyl-CoA. Notice the thioester bond linking the acetyl group to coenzyme A, utilizing sulfur instead of oxygen. Thioester bonds possess highly unstable properties. When this bond is eventually hydrolyzed, it will release a massive amount of free energy, enough to drive the subsequent reactions of the citric acid cycle forward. Because it initiates such a committed step, the PDH complex is heavily regulated. High intracellular levels of ATP, or an accumulation of acetyl-CoA, act as a negative feedback loop, inhibiting the enzyme and shutting down the gateway. The PDH complex functions as a strict toll booth. It only permits carbon to enter the matrix when the cell actively demands more ATP. Once past the toll booth, acetyl-CoA enters the citric acid cycle by coupling with oxaloacetate. It is commonly assumed that this cycle is a major ATP factory. In reality, a single turn of the cycle generates exactly 1 molecule of GTP, which is later converted to 1 ATP. This flowchart isolates the primary function of the citric acid cycle, the systematic oxidation of acetyl-CoA. As the carbon structure breaks down, high-energy electrons are transferred to carrier molecules. Each complete turn yields a precise output, 3 NADH molecules and 1 FADH2. You can identify exactly where this extraction occurs by looking for enzymes labeled dehydrogenase. These enzymes catalyze oxidation-reduction reactions, transferring hydride ions from the carbon intermediate to an electron acceptor. The citric acid cycle does not output immediate mechanical energy. It produces chemical potential energy, banked securely in these electron carriers. The speed of this entire extraction loop is controlled primarily by a single bottleneck, the enzyme isocitrate dehydrogenase. When the cell consumes energy, levels of ADP and NAD+ rise. These molecules act as markers of metabolic demand, allosterically activating the enzyme to increase its affinity for substrates and speed up the cycle. Conversely, when the cell is saturated with energy, accumulating ATP and NADH bind to the enzyme, inhibiting its action and slowing the cycle down. The entire carbon oxidation engine operates on a strict supply and demand logic, governed globally by the cell's shifting ratio of ATP to ADP. With chemical potential energy successfully banked, the action moves from the open fluid of the matrix to the physical barrier of the inner mitochondrial membrane. Embedded in this membrane is the electron transport chain. It consists of a series of protein complexes arranged by increasing reduction potential. This cross-section traces the energy's path. NADH arrives first, dropping electrons directly at complex 1. FADH2 drops its electrons downstream at complex 2. With each complex having a higher reduction potential, electrons are pulled sequentially down the chain until they reach oxygen, forming water. As electrons flow through this sequence, they release exergonic energy. Complexes 1, 3, and 4 harness this energy to physically pump hydrogen protons from the matrix into the intermembrane space. Complex 2, however, does not pump any protons. Because FADH2 bypasses the first pumping station at complex 1, its electrons ultimately yield less structural energy than those from NADH. This directional pumping isolates a massive, unstable concentration of protons trapped in the narrow intermembrane space. This gradient is defined by both a sharp drop in pH and a steep voltage difference across the inner membrane. Biologists call this a proton motive force. In practical terms, it is a literal electrochemical battery. To harness the energy stored in this electrochemical battery, the cell relies on a physical machine called ATP synthase. Protons rush back down their gradient into the matrix through the F0 channel of this complex. The sheer kinetic force of these moving protons forces the lower F1 turbine to rotate. This is chemiosmotic coupling. The physical rotation provides the 220 kilojoules per mole of free energy required to forge a new bond between ADP and an inorganic phosphate, generating ATP. But this tightly coupled mechanism is vulnerable to hijacking by chemical uncouplers. In toxic doses, a highly common drug acts as a systemic uncoupler, aspirin. An uncoupler effectively punctures the inner mitochondrial membrane. It allows protons to leak back into the matrix along their gradient without ever passing through the ATP synthase turbine. Compare this to the cyanide poisoning from earlier. Cyanide completely jams the electron transport chain. An uncoupler lets the chain keep running, but it destroys the battery's charge. With the turbine bypassed, the energy of the rushing protons is dissipated as extreme heat, presenting clinically as a severe fever. Oxidative phosphorylation demands two non-negotiable conditions to function: a continuous flow of electrons, and a highly impermeable membrane capable of holding a charge. With the mechanics established, we can calculate the final yield. Every mitochondrial NADH provides enough pumping force to generate 2.5 ATP, while every FADH2 generates 1.5 ATP. But there is a final architectural hurdle. The NADH created earlier during glycolysis is floating in the cytosol, and it cannot physically pass through the inner mitochondrial membrane to reach the chain. Instead, the cell relies on specialized molecular shuttles to hand the high-energy electrons across the barrier. This diagram contrasts the cell's two options. The glycerol 3-phosphate shuttle hands electrons off to mitochondrial FAD, dropping the yield to 1.5 ATP. Conversely, the malate aspartate shuttle hands them directly to mitochondrial NAD+, securing the full 2.5 ATP. This specific biological choice is why a single molecule of glucose yields a variable range of 30 to 32 ATP. The final thermodynamic output is not a fixed chemical equation, because in aerobic respiration, biological architecture dictates mathematical reality.